Hi everyone, thanks for having us here today. Just to let you know, our McGill team, we're presenting two separate projects that we've done, so we'll present them one after the other. My name is Jamie Schaefer, and I'll be presenting our Team 2 project with Jordi Volpe. And this project was of an oscillator coupled with quorum sensing and was the inspiration for our team name, Les Colis de Danse. Now, the inspiration for our project with the quorum sensing was a theoretical paper by Garcia-Ojalvo et al., where they coupled the standard Elowitz repressilator seen here, whereby tet-R represses Ci lambda, which represses Lac-I, which in turn represses tet-R and promotes an oscillation of the three genes. And they coupled it to a quorum sensing system using Lux-R and AI, whereby Lux-I produces AI, binding to Lux-R, which promotes Lac-I, and that feeds into the Repressilator system so that AI communication between cells as it diffuses in and out of neighboring cells should synchronize the oscillations in a cell population that all contain this gene construct. And in this system that they developed, Lux-R is expressed constitutively and is therefore always available to bind with the AI when it is produced. And just to note, the important part is that this is just a theoretical paper, and we were hoping to actually put it into practice. So we started looking for biobricks that would be available to do this. And one of them that seemed like a really obvious choice was I15004, as it contains a good portion of our quorum sensing system here with the Lac-I promoter controlling Ci lambda and Lux-I genes, and the tet-R promoter controlling the Lux-R gene. However, we still needed another component of our quorum sensing system, and so to achieve this, we ligated together R0062, S0100, and E0422 bricks to make a J40001 brick, which was the Lux-PR or quorum sensing promoter controlling Lac-I and with the fluorescence output of a degradation tagged ECFP. The final component of our system is the Elowitz Repressilator, available as I5610 with an output of YFP that is degradation tagged. We were hoping to ligate the Elowitz Repressilator brick with our other AMP-resistant bricks that we had ligated together already, so that we would do a double antibiotic selection of our cells. However, when we attempted to do the ligation, we had a problem with the digestion of the Repressilator in that it yielded fragments of the wrong size on our gel. We did, however, still carry out the ligation, hoping that it would work anyway, but there was no YFP produced in the cells, and therefore we're a little bit uncertain of the identity of the Repressilator plasmid as given to us. So we looked into the option of using other bricks, I5611 and I5612, which are both derivatives of the I5610 brick. However, the problems with those is that one has no fluorescent output to monitor the oscillations, and the other has the fluorescence output of YFP that doesn't have a degradation tag and is therefore not suitable for our oscillation period, which is about 45 minutes. However, we were able to experimentally test system 1, which is just our quorum sensing portion. In it, we had the Lux-I gene producing AI, as I discussed before, promoting, in combination with Lux-R, promoting Lac-I, which in turn represses Lux-I. This output was destabilized CFP, produced in conjunction with the Lac-I, and the quorum sensing was able to produce oscillations in this case. Our full system would have our quorum sensing portion coupled with the Elowitz Repressilator, as we before mentioned. However, there's a few differences between our system that we've developed and the system that we presented as our inspiration for the project. We have a couple extra feedback loops that are going on that weren't present in the other. The first is we have a Ci lambda produced at the same time as Lux-I, which wasn't present in the other system. This represses Lac-I, which in turn represses the production of Ci, creating a feed-forward mechanism. And we're actually hoping that through this, small changes in Lac-I as a result of quorum sensing will result in larger changes in Lac-I levels through amplification and would further promote the synchronization of oscillations in neighboring cells. Furthermore, Lux-R, rather than being constitutive in this case, is under the tet-R promoter and is therefore repressed when tet is produced. Now I'll pass it over to Horia, who will present about the simulations.
Thanks. All right, so to run simulations of our systems, we use the software BioNets. We've expressed the rates of reaction as continuum variables and the rates of transcription of the promoters as discrete variables. Most of the rates and assumptions that we have used in our modeling come from the Elowitz paper published in 2000. We have observed a few things when trying to model this. The first is that the models are very sensitive to the rate of transcription of the pLac promoter. This was not really addressed in the paper, and we're still investigating on the effects it has on the oscillations. Secondly, we needed a way to have the cells out of sync at the beginning. And it was unclear in the paper if they changed the protein or the mRNA lifetime. We decided to go ahead and have various differences in mRNA lifetime between our model cells. So it's only the more fortunate cases where the factors come together favorably to allow for oscillation that we see a synchronization between cells. In most cases, the biological constants create a lot of noise, and we haven't observed synchronization. But there are cases where it actually worked pretty well. The first simulation we have performed was actually to see if we can express our test system 1 in cells that express LAC-I naturally. These were the only cells that we had in the beginning, so we wanted to see if oscillations could still be observed. And as we see here, we have taken 2 cells that start more or less in sync, and in the normal case, they synchronize. And in the case where there is a LAC background, they also synchronize. The only difference is that the peaks are widened, and there are small oscillations here at this plateau. But this meant that the oscillation was still observed and that we could go ahead and perform tests and implement the system in these cells. Simulations for the model, that is the quorum sensing plus the repressilator, also gave interesting results. What we used is that we used a 3-cell system, 2 cells that are just regular cells, and 1 cell that has 10 times the output of protein as the other 2 cells. This is in order to have this cell represent an outside population of cells to which the other 2 cells will synchronize. So this mother cell, as we call it, is here in red, and we see that it's oscillating pretty regularly. The other 2 cells are in yellow and green, and this represents the levels of YFP output from the Repressilator system. Now with low coupling between the cells, that is, with low diffusion of AI from the outside to the inside, we see that no real synchronization occurs. They start pretty much randomly, and we cannot see any difference from start to finish. Whereas in the case where the coupling constant is strong, we see that the cells begin out of sync, and towards the end of the simulations, the peaks are more or less overlapping. So this means that actually the cells do synchronize with each other and with the mother cell. Now, the test system 2 is the model system that was created with the BioBricks. So it has 2 extra interactions, as was explained by Jamie. We have the CI here that is controlled by pLac, interfering with the repressilator and amplifying the concentration of LAC-I here by the feed-forward mechanism. And LuxR also, which instead of being constitutively expressed, is under the control of TET-R. So these 2 extra interactions gave a lot of noise, but also some interesting results. We've decided to test 3 sets, that is, with different strengths of the TET-R promoter, which is known to be pretty leaky. First one is with a tight TET promoter, semi-leaky, and with a constitutive TET promoter. At first, with no coupling, we see that the output from the Repressilator is oscillating, but is pretty much random. With semi-leaky promoter, also there are oscillations, but there is no synchronization between the cells. With coupling, there's also oscillation going on, but with the tight promoter, we can't really determine if these cells synchronize more than they did in the beginning. With the semi-leaky TET promoter, the cells show a hope of synchronizing here at the end. We can sort of see that the mother cell here with one of the cells has peaks that are sort of overlapping, as well as here. So there's a hope that this really means that synchronization is occurring between these two cells. The CFP output from the quorum sensing mechanism also oscillates pretty more or less nicely. And what's interesting to note is that when TET-R is constitutive, the oscillations are much nicer. This is the ideal case when we have the LUX-R constitutively expressed and not under the control of PTET, as we do, unfortunately, in our system created with BioBricks. So these are our results for the test system 1, that is only the quorum sensing part, which is a 2-gene oscillator by itself. If you look at these 3 cells, they're pretty much in sync with each other, but not necessarily with the rest of the population. And as time advances, you will see that the whole population of cell will become in sync. There you go. That's an on, and that's an off, and on again. This was done over a period of 8 hours with images taken every 5 minutes. And it's about 300 times the speed accelerated. Yeah, so that's, we observe pretty nice oscillations here in synchronization with the rest of the population. So that's encouraging. And we've done a bit of analysis on this video. Although it's hard to determine what the peak fluorescence of CFP is, we still managed to determine that the cells in green that you see begin in sync, and that as time progresses, the cells in red will become synchronized with the green population, and they will all oscillate more or less in synchrony with each other. And now back to Jamie for some concluding thoughts.
So basically what we concluded from our project was that we were able to observe synchronization with the quorum sensing portion of our system, and we also expect to receive synchronization when we finally construct the more complicated system containing the Repressilator. However, there's some difficulties with this. We will need to image CFP and YFP simultaneously, and we don't want to have any difficulties with interference between the two of those. And we also need to monitor the Repressilator alone as a control and just see to what extent or how much our quorum sensing system affects the synchronization of those oscillations. So now I'm going to turn it over to Adam, who will begin presenting about Team 1's project.
Okay, hello. So my name is Adam, and this is Alex and Julia, and we are Team 1 of McGill. So our project deals with bimolecular fluorescence reconstitution, and our work was inspired by an article, well, by a work that previously done by Professors Hugh and Kerpola of the Hughes Medical Institute. And this, they basically split a yellow fluorescent protein, YFP, into two sections that each, when separated, were lacking fluorescence when expressed as protein. However, if brought together in the correct orientation, correct conformation next to each other, they reconstituted their fluorescence almost completely, not completely. However, this reconstitution, like, they could not be reconstituted just in the cell. They had to be attached to other proteins. So this group, they attached them to two antiparallel leucine zippers that are transcription factors Jun and Fos that normally bind to each other and form a leucine zipper. So basically the C-terminal section of the YFP was attached to the Fos, and the N-terminal was attached to the Jun. So basically when expressed in the cell, they came and formed this leucine zipper and almost completely reconstituted the fluorescence. And this can be done in many variants, not just YFP, but different combinations of fluorophores can form different results. However, this work was done inside the cell, and what our team project, we wanted to do this outside the cell, and I'll pass it on to Alex.
Thank you, Adam. So we feel that extracellular fluorescence complementation could be very useful in the study of immunology, because as some of you may know, immunology involves a lot of intermolecular interactions between membrane proteins of different cells. And the fluorescence complementation allows a very easy visual confirmation of the interactions. To try this out, we needed to find a membrane-targeted protein that could carry our YFP halves to the membrane of the cell. So we found a paper written by the National Center of Biotechnology in Madrid. And what they had done is fuse the Fos and Jun domains to the IgA protease, the beta domain of the IgA protease of Neisseria gonorrhea. And they created two strands of cells that would bind together through the Jun and Fos and form lots of cell clumps, so they call them sticky cells. So what we did is create a fusion protein that you see up here. And this is the IgA beta domain that's fused to the either the Fos or the Jun leucine zipper and one of the halves of the YFP. So when the two cells interact, the Fos and Jun would bind together and bring the two YFP halves in the correct conformation to restore fluorescence. In order to make this fusion protein, we simply inserted the YFP genes in between the Fos and Jun zippers and the beta domain. And I'll turn this over to Julia to talk about our results.
Thank you, Alex. And now I get to do the fun part, which is to present our results to you. Thankfully, we have results in the form of some pretty pictures to show you. So just to quickly recap, what we were looking for was to isolate two separate populations of E. coli, expressing the two separate leucine zipper proteins, one population expressing Jun, the other one expressing Fos, on the cell membranes, and then in mixing them and coming together, the leucine zipper proteins would link, and the two half YFPs components would associate, and thus we would see fluorescence. And hence that's what we saw. Here you can see this is a bright field picture of Jun alone, and you see some of the islands and the coupling taking place, the sticky cells, basically, that Alex was referring to. And then here, on addition of Fos, you get fluorescence expression of YFP. Very nice. Here, this is something interesting that occurred. We had observed these geometric shapes, circular shapes, as you can see here. Let me anticipate your question about these shapes and how they formed with free-flowing bacteria. Basically, that is a point of further investigation for us. What we were mainly concerned with was the fact that YFP was expressed. And here, one hypothesis that we have is that because this was done on a slide, it was drier and stickier, and what might have happened is that the YFP protein, once expressed, was, as the bacteria were moving around, was ripped off of the bacteria and thus just stuck down on the slide, and this is why it's stayed in the configuration like this. I should mention that this approach was basically we had pre-mixed the two cultures and allowed them to incubate for about 90 minutes. And like I said, it was done on a slide which was drier. But another approach that we did was we used wet medium. We used water as our medium. Oh, I'm sorry. Let me allow to go back very quickly. Okay. Thank you. Okay, so here we have Jun alone in a patch pipette tip, which is on a scale of about 5 nanometers. And you have Jun alone here, and as you can see with the fluorescence filter, there's no fluorescence. Here you have Fos alone, and you have a little fluorescence here, but we attribute that to autofluorescence. And then here you have the two together. It's a beautiful expression of YFP here. Simply beautiful. And one other thing we wanted to do was to try to capture the actual reaction at the moment it happened, because it was possible to do this because the expected reaction time was about a minute, 60 seconds. And so using the same system of the patch pipette and the micromanipulator, we planned to squirt some of the Fos population into the Jun population and to capture it. So here we have the Jun population, all set it up, lots of cells here. And here under the fluorescence filter, Jun only, you can see, you don't see fluorescent bugs. But then here, once the Fos is added, you can see the fluorescence in practically a gradient fashion here with the brighter bugs because the Fos was squirted from this direction and less bright bugs here. And so that suggests another question of, what makes certain bugs more prone to this reaction and makes them express the YFP and thus be more fluorescent. And okay, so to conclude, basically, the future work that we would like to do is we'd like to make our plasmid into a biobrick through silent mutagenesis. We'd like to further explain this fluorescence pattern and the geometric shapes that I mentioned to you, and we'd like to determine the conditions and circumstances under which fluorescence forms and can remain stable. And with that, I would just like to acknowledge my team members here. That was a little dance move for you, because we're le colis de danse. And our ambassador, Andrew, and all our sponsors and all our supporters, some of whom are listed here. Thank you very much.
I would just like to add, because they forgot to say this, this is actually a very young team, so I'm very proud of them. All of the students here are second-year undergraduates. None of them were even of drinking age. And half the people here took the MCAT this summer as well. So they did remarkable work considering all they had going against them in terms of overwork and relative inexperience. And so I'm very proud of them. Thank you all.
Thank you. Oh, take questions? Yes?
First of all, I want to mention that it's really great good work for the team one. I have a question, where do you need to break this gene so that they can again reconstitute? You have this YFP8, YFP9, and YFP6. Do you have any idea where you exactly need to break it so that they can again reconstitute?
The YFP is a beta barrel, and the beta barrel is split right down the middle. And when the two halves are combined, the H bonds are restored around the fluorophore, which brings back our fluorescence. Yes. Does that answer your question? Yeah.
Any other questions?
Any other questions? Yes. Yes.
Well, we actually used biological constants for the simulation, so there was a lot of noise due to that. It wasn't like an ideal system also. We had 2 extra interactions that were not present in the model. And it's just overall noise due to various constants that were not properly like optimized for the system to work.
Do you think it's more likely to be better in the real thing when you build it, or worse?
It's hard to tell. It's certainly, there's a lot of factors that influence the frequency of oscillation in the cells, and we'll have to see, I guess.
Did it give you any insights into how you should build it?
We haven't really changed anything in our design based on those simulations. I think it's more showing us that it's very hard to do actually something this complicated into a living system. I hope that answers. Any other questions?
Before we ask that one, we're going to try to catch up a little bit.